on the foraging behavior and ecophysiology of common diving petrels. And the study is led by Brendan Dunphy from the University of Auckland. Is actually Brendan with us this evening in Auckland?
Brandon is with us. Oh, there he is.
Yep. Oh, yes. Yeah. Good evening. Thanks so much for joining.
Oh, you're welcome. Kia ora koutou, all the way from Aotearoa, New Zealand.
Hey, nice to have some Kiwis with us.
Very nice.
Actually, I see a few Kiwi faces on here this morning.
And so we picked this paper because we thought that it was really important to gain less known seabird species. That's very much along the line of the pelican paper we had last week, and also diving petrels are.
Oh, we may have lost.
Felt that it was important to gain more understanding of their ecology. You losing me, Grant?
Yeah. I was just going to say, I think you might be cutting out a bit there, David. You might have to cut your camera, I think, to save your bandwidth. Oh, and with that, David has disappeared. Oh no, he's back again. Can you hear us all right, David? Oh, here we go. Technical difficulties. This is the first time this is pretty good in the 8 weeks of doing this. This is the first time we've lost David like this, considering he's in the middle of nowhere in France. Yeah, I think his garden link is a bit weak. So with with that, I do have some of David's notes, which I could probably read a little bit. Just stand by a second there. I think he sort of prepped me for this because he knew that he was probably going to have some issues this morning. He's just recently moved houses again, and it's always a bit of a run to figure out what his internet connection is going to be like. All right, so this paper here, so I managed to get around to skimming over a little bit of this paper last night in my haze of trying to do some coursework that I'm in the middle of at the moment, and I thought this was quite interesting. There was quite a dichotomy in this paper that came out. You're looking at these two different populations of diving petrels that are very close to each other. They seem to have very different ecologies, and from what I gather, the colonies are about 50 kilometers apart in the Hauraki Gulf. Is that right? Oh, here we go. Here's the beat.
Yeah, I mean, they're about 70 kilometers if you want to be precise, but yeah, they're very close.
Yeah, and yet you still have, it's quite funny because they seem to be foraging in completely different trophic levels, despite being so close to each other and obviously being the same species. It's totally wild. But oh, David, can you hear us?
Yeah. Okay, but carry on because this had to happen at some stage. My network is very unstable this morning.
Yeah, it might be a good idea, David, to cut your camera. It might save your bandwidth. Oh, very good. Anyways, so Brandon, so anyways, it was quite interesting. These birds are obviously occupying different kind of ecological niches, and what I find really interesting about that is, especially just because my understanding of the Hauraki Gulf is that even though you have patchy prey distribution, I wouldn't have expected the same species in the same area to occupy such different trophic levels, or to be foraging from such different trophic levels. Do you have any indication of what might be driving that? Is this I know in the paper you sort of touch on it a little bit, but is there any reason why is it because they're just not flying as far? They're not overlapping in space.
No, they're not overlapping in space. And what we actually see in the Hauraki Gulf, if you sort of do that with your fingers and then just draw a line, that's kind of like the Hauraki Gulf, and the two colonies are there and there.
Yeah.
And what we find is near Auckland City, I'm sorry for those of you that aren't familiar with the Hauraki Gulf, but at the base of it is pretty much our biggest city. Near there, it's the predominant zooplankton species or groups are your barnacles and your bivalve larvae, which aren't really preferred prey for the species. It's sort of feeding more on copepods and krill. And I think that was really what drew us to work on this bird, is like, well, what's going on with this species? Is there any evidence of sort of small spatial scale differences in what they're feeding on, how they're behaving, how are they interacting with the environment? And, we were quite pleased to be able to pick that up. And so out towards our other colony, the Mokuhino Islands, where there was a lot more higher density of, traditionally high density of krill and copepods, and we just see they're not working as hard. They're not, it's a much easier life basically for these birds out there. And so that was really exciting.
Yeah. And I assume that they're even when they're breeding, for example, they're occupying similar, the conditions on the islands are relatively similar. There's no difference between the sort of nesting habitat or anything like that. Is that right?
It's not hugely different, no. I mean, both of them are sort of recovering islands. They're both predator-free.
Yeah.
It's digging into the soil, burrow nesting. So, yeah, I mean, both of them were farmed previously. So there's slight differences, of course, but not massively different. Maybe some of the other Kiwis on the audience would disagree, but it's been hugely different.
Yeah. It's kind of a shame we've lost David, because I know this is sort of one of David's favorite topics. Certainly, he's always had some opinions on cortisol type studies, but he also loves diving petrels. I know he's done some work with on diving petrels in the past as well. I believe you actually cite one of his recent papers at the end of your study. I
can actually hear you.
Oh, hey David, there you are.
Yeah. Yes, sorry. Yeah, I switched off the video. I had some network issues. This has to happen when I change places each single week. Yes, so thanks a lot. I really enjoyed reading the story, and I guess the point about the corticosterone aspect was that you had really contrasting results depending on the breeding phase. With for some phases corticosterone being higher at the smaller colony and then lower, and this was quite hard to interpret, I guess.
Yeah, it was, particularly in the weight data itself didn't really lend us any clues either. So just sort of, it's fascinating, of course, but it makes it, as you say, a harder story to tell. But we did find, a significant increase in corticosterone secretion during chick rearing. So there is that sort of suggestion that as they're having to provision their chicks, is a much more stressful time for them.
Yeah, because you put forward the indicator aspect of the species. And also the fact that you want to understand the constraints set on the different colonies by the marine environment and potentially different prey types and all that. And I was thinking, there might be other ways to get at it than measure corticosterone levels, which also get influenced by many other things. I was wondering whether in the end, for instance, if you would manage to put accelerometers on the diving petrels, whether you would get a much more direct proxy of how hard they are working. And these accelerometers now, they are even smaller than the GPSs you put on, so it would be practicable, and maybe then you would get a stronger metric than corticosterone level.
Yeah, well, it's funny you should say that. Watch this space because Kyle Elliott is going to come down for a sabbatical next year, and we're going to do exactly that with accelerometers and some basically working out some daily energy expenditure data as well. So, yeah, hopefully that will be a more high resolution, more a story and a better indicator coming from that data stream. So, fingers crossed.
Yes, and I missed a bit of the discussion just before, so sorry if this repeats some of the points you addressed, but it struck me also how coastal the species is, and I felt that probably for an oceanic seabird, it was interesting to see them so coastal and actually handy in terms of, for instance, designing marine protected areas. Could you maybe comment on that?
Yeah, I think what's driving that is the lack of a proventriculus potentially, and so they can only do daily foraging trips, and so it limits their range from the colony compared to other procelariforms, which can do their 3 to 4 to 7 however long day trips. So I think in that respect, as an indicator, it's fantastic that little tweak of biology works in our favor, and that it limits them as to where they can go.
But overall feeding conditions seem to be quite good for both colonies, or at least the productivity data you showed indicated that.
Yeah, in terms of, it's a pretty raw measure, chlorophyll A, but we didn't really pick up much difference in it. It's potentially just the scale over which we're working. So in terms of productivity, yes, but as I mentioned before, there are differences within the Hauraki Gulf. So our Gulf waters towards the city are more dominated by preferred prey species such as barnacles or bivalve larvae compared to the outer Gulf colony, which is more dominant with krill and copepods. So potentially a richer feeding area for the species.
Mm. And the contrasting signature in terms of carbon isotopes, and not only nitrogen, there was also a differentiation for carbon. Was it more something like an inshore offshore gradient in the carbon signature, or how do you interpret?
Yeah, that's how we sort of interpret it. Yes, basically.
Okay. And then the higher trophic status for the smaller colony pointing to birds feeding more on fish larvae, right?
That's our suspicion, and that's really what we want to follow up next. Is what exactly is it? I mean, the beautiful thing about New Zealand is there's a lot of species here, and there's not much known. So research-wise, it's fantastic, but it's also frustrating in that it can limit sort of what your conclusions you can draw. So yeah, that's really where I want to direct some students into the next phase is actually quantifying the dietary elements and finding out what specifically are they feeding on. And it's how much proportions in the diet, and just get a real good feel for what the birds are actually feeding on. Admittedly, ours was a snapshot of what they could potentially do. So we just want to again up that temporal element to it, and also quantify more and high resolution information about the diet itself.
Yeah, well, thanks for opening a window into the world of these common diving petrels. As Grant said, there are lots of common features with little hawks, so that was also a reason why I got excited about the work. And with this, I'll hand over to Grant for the question.
Yeah, actually, you've kind of already touched on my question, which was sort of where we're going next with this, and how we quantify the actual diet, and whether or not we can show what they're specifically eating. And is there any, I don't know, I mean, not being a physiologist or anything like that, or knowing anything beyond my small sphere of knowledge, is there any indication that is there any actual indication that diet influences cortisol levels? Is there any evidence for that in the literature?
Yeah, there's a fair amount, and I tend to feel some people push it too far and say, you kind of come away with the conclusion it's the only thing. But there is a pretty good body of work showing that diet, and particularly around low food levels in the environment, can influence court and lift it as the birds are trying to just maintain homeostasis as their condition drops. Mm-hmm. So the linkage between that's quite good. But there are other factors at the colony that are difficult to account for. It could be competition for burrows. It could just be, I don't know, mm-hmm. Something scared them or whatever.
Yeah.
It's a great measure, but it is very sensitive to things other than what you're trying to detect.
Yeah. I wonder if you could talk a little bit too about the relationship between cortisol and the size of the birds. I know you mentioned that there's a hypothesis out there that there's some negative relationship between cortisol levels and size of birds. So are we in a broad sense, does that mean that larger birds tend to have lower overall background cortisol levels? Is that my understanding of it?
And if you mean by larger and better condition, yeah, they do. Mm-hmm. But those that are in poor condition and starving, they will basically pump out a lot of cortisol, release a lot of glucose into the blood, and just try and cope with these sort of low food starvation conditions, which is fine in the short term, but over the long term can have really chronic maladaptive negative effects on any species.
Yeah.
Just included. So.
Yeah, of course. What about say between species, so interspecies relationships between cohorts? So for example, are there any marked differences between, say, albatross and the diving petrel with regards to cortisol levels? Is there any sort of relationship across the taxa?
Not that I'm aware of, and it gets people are a little reluctant to compare court and interpret court among species. Like high court for one species may actually just be baseline. Mm-hmm. So we tend not to we'll place too much emphasis on what court means among species, purely for it is so species specific as to how it acts and how you interpret it and what it actually means. Yeah. So that's potentially a limitation of the method, but that's pretty what we've got.
So there's been some recent work in Antarctica looking at cortisol in guano samples. Is it comparable to use guano samples versus blood samples, for example, or is blood sort of the best way forward? What I'm trying to get at here is, are there less invasive ways to get cortisol?
Yeah, there are. And so blood, it is the best, but it is quite invasive. Guano and blood will sort of reflect what's happening over the last hours on that scale, whereas guano is sort of in the next sort of day, 2 to 3 days, I understand. I haven't analyzed it myself, but my understanding is quite variable. My other issue with it is there's the potential then to be measuring the stress hormones of the prey as the basically preyed upon, depending on the hormone you're looking at. But the one we're working up now is actually feathers, and I saw carriers online who've done some feather stress hormone in little blue penguins, and I mean that's just fantastic. We can take feathers that have been fledged from a chick, so we don't even have to touch the chick, and analyze what was the stress of that bird as that feather was formed, and then that allows us to compare among years, and so we can start to identify poor years and unpick what that might be causing that. So that's sort of where we're heading next. I am of the opinion that the less we can handle the birds, the better. So other samples such as guano, feathers, fantastic, depending on the question you're trying to ask.
Yeah. And actually, I do see. You're talking about Carrie Lukey's? Is that who you're referring to?
Yeah.
I don't know. Right. Yeah, that's really cool stuff. David, do you have anything else for Brendan before we open it up to questions?
No, but I'm happy we touched on the many caveats to an approach based on corticosterone levels. As Brandon said, it's highly sensitive, and in my opinion, it has been misused a lot. And I've seen actually quite a few papers with corticosterone levels going all over the place, and then people then trying to interpret. And in my opinion, the corticosterone level is really a byproduct of metabolism, and hence, if you want a strong indicator of how hard an animal is working, you'd better look directly at metabolic level. I think that's the way to go.
Yep. Yeah, that's good stuff, Dave. All right, so let's open it up to questions from the audience. We've got one from Adrienne Gall has typed one into the chat, and she asks, what physical oceanographic features might be influencing the selection of foraging location and prey availability at those locations?
It's on such a spatial scale that we don't actually get fronts. We're out towards the end of the outer reaches of the Hauraki Gulf. We've got an upwelling around the Mokuhino Islands, and they actually rise up, and so they can bring up nutrients and cause ephemeral plankton blooms, etc. So the Gulf itself is very patchy, but it's very productive. It has its own resident population of Brooder's whales. So it's a small area, but it can support 70 plus whales year round. So it is very productive, and obviously with a city quite close by, there's actually a lot of farming. There's a lot of nutrient flown into it anyway, which is adding to the productivity, and perhaps not the best way. So the birds themselves aren't really, from the snapshot we saw, aren't really associating so much with features. The environment itself seems to be sufficient to support them, but we really like to look at that further, of course.
Yeah, it's just surprising that you would have a difference in trophic level feeding in an area that's seems like it's not all that large, if there weren't factors affecting what's available close versus far or something like that. So thanks.
And actually, just following up on that, it is an interesting point. But I think if when you look at there's a figure, I think, where you show the overlap of the tracks with the oceanography. Yeah, here we go. So figure 1 in your paper here shows that there seems to be some sort of, there's out near Burgess Island, it tends to be more of a slope associated with it. How steep is that? Is that deep enough out there that it would you'd have a completely different sort of prey structure? Or is that just something that's not as steep as it actually looks in the figure?
It's fairly steep. I can see Chris Gaskin doing this, so
yeah. Maybe Chris has a word to say. No.
But we do see as we mentioned, we do see differences in the zooplankton communities out there, and that is a year-round pretty common thing that we see. So that's pretty stable through time.
Yeah.
The composition of the species, anything like that, will change, but that differences between the two areas that I've mentioned, bivalve larvae and krill copepods, tends to stand. So,
yep. Oh, we do have K Milton is raised hand. Here, I can unmute you, and you can speak out loud rather than type.
Yeah, okay. It's actually John Stewart, not Kay Milton, but never mind. Kay's sitting beside me. It's her computer.
Yeah.
I was just pointing out that the colony on Tiri Matangi may not be much smaller than the one in the Mokuhinos. Back in the late 1980s, Graham Taylor was there, and he thought there might be 4,000 pairs on Wooded Island, which is about 200 meters away, possibly up to 10,000 pairs. The population's growing rapidly on Tiri Matangi, but we don't know what's happening on Wooded Island. So actually the Tiri Wooded Island population may be quite significant. and not a lot smaller than the Mochahinau's one, and so the density effects and things like that may not be so different.
So I might need to put in a correction on the paper then, John.
You sir, yeah, second edition.
Yeah, yeah.
There's always room for volume 2.
Yeah. I'd just like to take the moment to thank John, because him and I sat out there attaching tags to these birds, and he was quite instrumental in work in the rain and thunderstorms. So thank you, John.
You're welcome.
Thanks, John. All right, do we have any other questions from the crowd? I don't see anyone desperately raising their hand or waving at me. All right, so with that, I will thank you very much for joining us, Brendan, and thanks for a cool paper. I do love diving petrels. They're I spent some time in my PhD on Whenua-Ho doing my working with cities, and so I had was often dodging the South Georgia diving petrels that breed on the island down there, and certainly spent some time on South Georgia as well. And yeah, they're absolutely stunning little birds. So thanks a lot for joining us. And I think with that, David, I think we can move on to the penguin paper.
Yes,
Thank you very much.
Cheers, Brendan.
Yeah, thanks a lot. And I'll try to present the second paper, and if my internet messes up again, you'll wave your hands, Grant, and
Yeah.
or probably I'll see everything freeze again.
Yeah, I think you should be okay. I can hear you pretty well now, and you're unbroken. So I think we've got Teresa with us this morning. Is that right?
Yes.
Yeah, there she is. Oh yeah, okay. She's waving at me. I can unmute.
Excellent.
There we go. Hi, Teresa.
Excellent.
Hi.
So we will discuss the paper you're co-signing with Hailin Pan from BGI Shenzhen and the Chinese Academy of Sciences. And TESCOL is based at the University of Otago. And their paper, along with many other co-authors, is on high coverage genomes to elucidate the evolution of penguins. And it's a really interesting project which explores the past to understand the future by using phylogeny and the evolutionary history of penguins. And the fascinating point, of course, about penguins is that they've been around for over 60 million years. And in comparison, we're really newcomers, and that always reminds us how much we've messed up in this short period. So demographic models have demonstrated dramatic penguin population declines during the ice ages, followed by rapid population expansions in response to global warming. But future global warming is predicted to cause significant population declines, and genomic studies are thought to help us forecast these future dynamics. Now we have 21 penguin species, as you know, but there were actually over 50 of them, and there are over 50 extinct penguin species in the record. Just think about it. And today's penguins span a modest range size, with the emperor penguin at 30 kilos and little penguins at 1 kilogram. In contrast, the fossil record has penguins taller than me, which is not so difficult, and weighing over 100 kilograms. So imagine yourself trying to fit one of them with a GPS tag.
You could put a brick on that. You could have a brick as a GPS tag on one of those.
With a nose, it has to be streamlined.
Oh yeah, of course.
Yeah. And previous molecular analysis disagreed on whether Aptenodytes, so that's the emperor and king penguins, or Pygoscelis, that's Gentooth, Adelis, and chinstraps, or both together represent the sister taxa to all other extant penguins. And but they suggest a polar ancestral area for extant penguins. In contrast, the fossil record suggests that the more temperate adapted genus, like Spheniscus, so that's African, Humboldt, Magellanic, and Galapagos penguin, was the first to diverge. Only a few studies have explored genome-wide evolutionary processes among penguins, and these studies have focused on just 2 Antarctic taxa, the Adelie and emperor penguins. Now, the authors of the study we're interested in present 19 new genomes that, together with the 2 previously reported genomes, encompass all extant penguin species. So they present this with a whole armada of techniques and technology, very impressive. And sometimes in the methods, it even becomes poetic when they talk about supernova assembly. I really love that term. And they obtained almost complete high-quality genomes, which on average, with an average of 90% complete core genes for each of the 21 penguin genomes. And the information is now available to the research community, for instance, to explore penguin adaptation and population persistence, and then the incidence of global changes. And if you look at the figures of the paper, I think it's figure 3 is really nice because it's a constructed phylogenomic tree, which in contrast to previous studies suggests that Aptenodytes, so emperor and king penguins alone, is sister to all other penguins. And hence this points to an Antarctic or subantarctic origin of the penguins. So congratulations on this paper. And my first question would be that indeed the paleo record gives a slightly different story, with Australia and New Zealand actually being the cradle of all penguins. So now we're shifting to a story with a stronger emphasis on Antarctica. Is that right?
Well, not entirely. So there are different approaches that have been used. So previous papers using fossils have indeed showed New Zealand to be have the oldest penguins, and I know Alan's on here as well. He's authored a number of papers on extinct penguins, and I think it's pretty clear still that New Zealand is probably the place where penguins evolved, but that's in deep time. In this paper, we're more interested in looking in more recent time. So when did more recent penguins split from each other? And Antarctica is definitely an important part of the puzzle there.
But I always think that in Antarctica, so much is still buried under ice, so to say. So could this be masking a whole part of the story? Again, if we look more at deep time?
I suppose it could be. There are fossil records of penguins throughout South America. I'm not a paleontologist, so I'm probably not the best person on this group to comment on that. But I think that there's numerous more fossils to be found, and there's lots of scrappy fossils that have already been found but haven't even been described yet. So I think in terms of the evolution of penguins, we're going to be looking more in New Zealand and maybe Australia, South America for the time being, but look in maybe when global warming continues, Antarctica will open up some more fossil beds. I don't know much about the geological history back there.
No, that's quite fascinating. And but in terms of genomics, so and for people who are not into genomics in the Seabird audience, once you have a genome, how exactly do you proceed to test, for instance, penguin adaptation to forthcoming warming events? How would this work in practice?
Well, I guess at this stage we're looking at a few different analyses. I've got them under 3 broad umbrellas in the paper. The first one is the evolutionary history, so looking at the phylogeny of penguins. So when did they all split apart from each other, and why? And in some of the previous work that I did during my PhD with a bunch of other co-authors, including people here on the meeting, we found that geological island emergence has likely facilitated some evolution of more recent species. So we'd like to use the genomes to look deeper into that and see whether that's occurring with more clarity and better statistics. So we're just using better data to address some similar questions. And these questions have been asked for a long time by many, many different people. Another way we're using the genomes to understand these adaptations to climate is using demographic models. And this is just a way of seeing the population size over time. So in previous papers, we've been able to show that populations have fluctuated with different ice ages and other cycles. So you can look at the last couple of thousand years, maybe 20,000 years, quite well with the data that we've already got. But with the genome data that we've just published, we can actually go back 1 million years or more. And that will give us a lot more resolution about how these birds have adapted to, warming events and cooling events during the Pleistocene, and then be able to relate that to scenarios in the future. And that's going to be using some fancy modeling. And then the third way of looking at this adaptation is by using the genome and actually zooming in on certain sections of the genome, which will give you actual genes, and those genes may be related to temperature or whatever else you want to look at. Maybe it could be about insulation and feathers and the development, and you can use those information and check between all the different penguin species and other birds and see which ones have the genes under selection for this temperature. I hope that sort of answers the question.
Yes, thanks a lot. It was very clear and very helpful. From a methods point of view, you speak repeatedly about high quality blood samples in the paper, and I was trying to figure from a practical point of view in the field what high quality blood samples would mean, also in terms of logistics involved to collect these samples.
Okay, so I've worked a lot on ancient DNA, so this is really not high quality bone samples that have been collected, over the last 100 years, and those bones may have been lying in the ground for 1,000 years. So the DNA over time gets degraded, and that happens for a variety of reasons. Bacteria can break down the DNA. You can also have UV damage, or just temperature can also break it down. So museum samples also are prone to having damage. So for this project, we really wanted to get the best quality sample we could get. And to do that, we did our very best to collect blood samples. And that was, most of these samples were collected by all sorts of people. It was a huge collaborative effort to do this. Some of them were collected only 2 weeks before we got them, we did the sequencing, so they're very fresh. And other samples have been collected maybe over the last year. So in general, they're pretty good in comparison to bone samples or museum samples, which you'd otherwise get a lot lower quality. And I guess one example that's important for this project is we have recently expanded our data set, and we've been trying to get genomes from some extinct penguin species. And those species are about 500 years old, maybe 700 years old. And the DNA is so degraded that we have been able to construct a genome, but it's got a lot of missing parts to it. So for the analyses, we won't be able to do all of the analyses with those genomes, but we'll be able to get a better understanding of what's going on than otherwise.
Yes, and how much blood is needed for this type of analysis for each single sample?
Not so much. We've been collecting maybe up to a mil of blood per bird. Okay. Yeah, so not very much. This is pretty standard, I think, for most seabird researchers, is to collect about one small vial. And I think that even for the extraction, we might not use all of it. Yeah, it could be already a lot for much smaller seabird species, but I guess for penguins, that's probably all right. It could be. I think that less blood is still okay. So this project comes under the umbrella project of the Bird 10,000 project, which I'm not sure how many of you are aware of it, but it's a project to sequence the genome of every single bird species in the world. So 10,000 birds. And of those, there are some tiny birds. And to do that, they are using smaller quantities of blood and tissue, and some museum samples too. They'll get what they can get, from many of these. And is there, so if specifically on the penguin project, if
if folks still want to join the consortium. Is that still possible?
That's still possible, of course. I recently was awarded a Marie Curie Fellowship to work on this for the next couple of years from the University of Copenhagen. And the next year I'm going to aim to spend in New Zealand, and I'm going to be collecting more samples or collating more samples. So I'm more than happy to involve anyone that would like to be involved in this research. One thing would be great to start to get population data sets, so not just looking at one individual from each species. It would be really great to start looking at populations, so maybe different islands across the Southern Ocean, and looking to see whether there's different trends going on in the demographic modelling and adaptation with those. So please send me an email if you'd like to be involved.
Yeah, very nice. Yeah, congratulations. Grant, do you have any questions?
Yeah, just quickly, Teresa, I wanted to follow up on that last comment. There's and you might already be aware of this, but there are people who are working on collecting blood samples for different reasons in Antarctica at the moment.
Yep.
Gentoos and that's. I don't know if you've already been in contact with the folks at Stony Brook University, but well, if I remind myself to do it later, I'll drop you an email and put you in contact with them if you haven't already.
That would be great.
There's also quite a reasonable spread of data as well, because I believe they're collecting data from a whole pile of different islands from different species.
Fantastic.
So you might get some.
I am involved in research with Tom Hart, who works at University. Yeah, so he's been providing some samples, and we've got lots of Gen 2 samples already, and some more genome as well to add to the next follow-up paper, which will have the results or some of the results and analyses in there.
Yeah, that's cool. Yeah, I know Tom really well. I was actually just Skyping with him over beer yesterday.
That guy.
Yeah, that guy. So anyways, the what I really thought was cool about this paper is that it doesn't really follow what I would call a typical the typical structure of a paper. You go in, there's a lot of detailed methods, and a lot of it really seems to revolve around you've got this data set, you've wanted to just get it out there to start, and then invite people to join in, rather than just being, here's a paper, here's what we did, and isn't this cool? But this is really an invitation to dig into some really interesting questions around penguins, and have not only how they're sort of sentinels of change, but evolution in a broad sense, and how we can answer some really interesting questions around that. As David had said, and we've seen before, we've mentioned before, these penguins have been around for 65 million years, and I mean, if you go back 65 million years, Antarctica was still temperate. Like it wasn't Antarctica 65 million years ago. So whether or not they were in New Zealand or Australia when they branched off, they were still in sort of they started in sort of a temperate climate, and then as Antarctica shifted south about, I guess it was about 25 million years ago that it finally got covered in ice. These birds would have already gone through a whole slew of different environmental conditions, and yet there's only 50 species, or 50 known species that have ever existed, which seems interesting to me because of the variation and how long they've been around.
Well, I highly doubt. I think there's a lot more than 50. Sorry to barge in there.
Yeah, please do.
I'm sure there are many more than 50, and they keep being published every year. There's a number. There's more penguin species being published from from the past, and it will continue to be that way. For, a long time.
Yeah, and that's great, because I think that goes back to how we can answer questions about how these animals are evolving to deal with different climate regimes, and certainly relates to where we're going now with regards to how fast things are changing now. I reckon, and maybe you can speak to this a little bit, is how do we go about elucidating whether or not these animals are going to be able to adapt, and will they adapt evolutionarily to the changes that we're about to see in the next 50 to 100 years?
Yeah, I would really like to address that too, and that's one of the questions that I have proposed in my fellowship application. So I'm hoping that I'll be able to follow that up. It's, I guess, all about the methods that you use, and I'm not very strong with the modelling at the moment, so I will have to do a lot of learning. But I think using the demographic side of things is going to be very important. So part of the project I did during my PhD, which this genome project kind of bounces off a lot of my research that I did in collaboration with many of the same authors, was to look at this ice age kind of fluctuations in population size, and as well as spatial kind of distribution of penguins through the Southern Ocean. And so when it's very cold, you see that penguins have to expand out to the further north, and then when it gets a bit warmer again, they can go back down towards the poles. And so I guess that's one way of kind of looking at how climate may affect them. So as it gets warmer, I guess we can continue to go south because they are adapted to being in cold water and not being in particularly warm places, except the Galapagos penguin, which is a bit strange. But in general, they all seem to be polar or Southern Ocean cold adapted. I'm Australian, so I think everything in New Zealand is cold and further south. And so I think looking at those kind of big broad patterns and then trying to use that data from the genomes from the past to try to model what is going to be happening with future climate modelling may be one way of doing that. And I'm going to need to get some modelling experts involved in this project when it comes time to doing that. But talking about the fossils, when you were just talking about that before, another point is that one analysis that we are doing at the moment is we're combining the genome data from all of those species that we've got, as well as fossil data. So we've actually got all fossil and extant penguins in a phylogeny, and you can use that information to then understand how geological and climate events may have influenced their the evolution and when they split from one another. So this is unpublished work, but this is something that's really cool that we've been getting some early results on. And it does look indeed like New Zealand is probably the place where it all happened.
Yeah, really interesting. The last point I wanted to bring up, which you discuss in the paper, you got a section on the cultural significance of penguins, and certainly in New Zealand, as their penguins in New Zealand are treasured possessions, and there's a lesson there, I think. And I'm wondering maybe if you could talk about that a tiny bit with regards to how those lessons might be translated to the rest of the world. If the rest of the world viewed penguins as treasured possessions, do you think that would do you think that there's a way that we can translate that sort of cultural significance to the rest of the world?
Yeah, I definitely there's a lot of points to bring with cultural heritage, and New Zealand is at the forefront really of bringing culture into research, and it's present in everyday life with language here and with politics, and it's really great to be in a place where it's valued so strongly. And so I think that it's a lesson learned for everybody, including myself. This is early days with learning about permits and learning about permissions and agreements, and so everybody can learn from this. But I think that in terms of using it in other places, if somebody in Australia wants to work with penguins, it's just to collaborate with people and ask first, and ask for what would be the best way to approach it, and what's valued under those cultural perspectives. And I'm talking about other indigenous peoples around the world as well. South America and Australia would be nice. to have that included. But you know, this is going to be a political talk here, so I'm not going to
Yeah.
dive into that too much.
Yeah, you gotta be careful.
And of course you can totally transpose and develop upon this, for instance, in the Arctic, with Arctic First Nations. It's exactly the same context, and as Grant, I did enjoy very much this part of the discussion.
Thank you.
Yeah, it actually would be quite nice, I think, in a lot of in more papers to see this sort of section built into it. I mean, I think it's really important that we're able to put seabirds and well, any species we're working with really in context of the people that are around it. Oftentimes, and probably most of you have experienced this at some point or another, that seabirds and marine animals are out of sight, out of mind for most people. There's not really a clear way of connecting. You know, someone in the Midwest of the United States or Canada, for example, who's never even seen the ocean before, has no indication of how people relate to these animals. And so I'm really was really excited to see that statement in there, and it'd be nice, I think, as a template going forward, if statements like this were put in papers in general to try and bring these birds around to people.
Thank you. Yeah, I agree. It would be, and it's very nice to also start to include people in the work, because I think that there are so many new perspectives that you can get from different cultural backgrounds as well. And these are all going to be important for future grant writing as well. Which, you know, this is going to become, it is already important, but it's going to become a lot more useful and meaningful for somebody in the Midwest of the States, you know. Yeah.
Well, thanks very much. All right, and with that, I think I'll open it up to questions from the audience. If there's any burning questions out there, please raise your hand or wave frantically. I don't see anybody immediately. David, do you have any other discussion points? Oh, actually, we do have one. Here we go. I'm going to, good. You've already unmuted yourself. Go for it, Alan.
Hey, this is the first one of these I've joined, so might have to join some more, Grant.
Welcome.
Thank you. Thanks to the speakers already too. It's very interesting. I just wanted to make a comment really about the fossil stuff. So that there have been, Antarctica is one of the richer places for penguin fossils actually, but it's only in a limited time zone really. So the Seymour Island area, you know, the ice-free areas up the peninsula, has got you know, an incredibly rich fauna, but that's from like 40 to 50 million years ago, and so that's showing that there was a really big diversity of penguins at that time. And from New Zealand, we've got sites that are around 25 million years ago that are showing incredible diversity as well. And even in the older period, around 60 million, there's a huge diversity of penguins at that time too. So it seems that penguins have been able to reach quite high diversities in small areas in all parts of the southern hemisphere, I suppose, over time. But of course, you know, the work that Tessa's been doing, being on genes, can't really tap into all that old stuff. But so this is all just talking, all this discussions about the crown penguins, all the recent groups. But it looks as though in the past, you know, the older diversities of penguins were doing all sorts of other things. So they were getting bigger and they were living in warmer climates. And so now we're just left with one branch, which is a more sort of cool adapted group of penguins. So yeah, there's quite an interesting history going back, which doesn't necessarily reflect what the modern penguins are doing. But penguins are also fortunate from the fossil record that they're very heavily built birds, so they're actually some of the best preserved marine fossils in the southern hemisphere in terms of vertebrates, or certainly in terms of birds, they are the best. So we're quite lucky with penguins, and as Tess says, there's going to be plenty more fossil penguins described. So one of the rules of paleontology is that basically what has been described is only a fraction of what was there in the past. So while there may be only 50 kinds of extinct species described, there were no doubt hundreds or thousands of them if you actually go back 65 million years, and we just got to discover them still. So that's all I wanted to say, really.
That's interesting. And so, just out of curiosity, like, you know, we've had what's caused the extinction events? Has it been ice ages? So is it the glaciation that's pushed these previous groups of penguins out, or is it something else?
I don't know if anyone could answer that, because there's not, even though there's a good fossil record, it's not that fine enough that you can see what happened between 40 and 39 million years ago.
Yeah.
Or that would take a lot more data, I think, to figure out. But yeah, obviously, well, I'm presuming temperature change has affected things in the past. That seems logical, but I don't know how much evidence we have to say so. I mean, if you look at size variation, there was more size variation in the past. I mean, you hear about all these giant extinct penguins way back in time, but there were also quite small ones. So you only hear about the giant ones because they're more impressive, but there was actually more diversity. So what's actually caused that change? I mean, one of the theories is that the big penguins have gone specifically because of the evolution of mammals which might have either outcompeted them or eaten them. So if we go back beyond 25 million years, there were kind of no seals, for example, and a lot of the odontocetes and things that are younger than that as well. So there's basically the mammals have taken over a lot of the niches of some of the bigger that maybe some of the bigger penguins occupied in the past. So.
Yeah, I mean, I think it's so fascinating that these birds have been around as long as they have. I mean, 65 million years ago, the dinosaurs were just reaching the end of their rope, and these guys seem to thrive and expand well beyond many other a lot of other species that got totally hammered at that time. And then, so what I was really trying to get at there was whether or not there was any way of elucidating how those changes in old populations, if there's any way of figuring out whether or not, or how that might translate to what's going to happen to penguins in the next, say, 100 years or so. And I think that yeah. But anyways, so that's very cool, and thank you very much for joining us and helping with that fascinating discussion and very cool paper. David, do you have any more?
Oops, I'm unmuted. So I guess the whole point is also that penguins and other seabird species might actually cope much better with global warming than we think, just because also they've been through so much in the past, including warmer periods. So that's a kind of way to put things on their head, I guess.
Mm-hmm. Yeah. Maybe we've already stuffed them up quite a lot. So if penguins have to be pushed further south and there's no, they get to the beyond where the subantarctic islands are and they can't actually go any further, then I guess that's going to be a bit of an issue with what we've got left. But yeah,
certainly in Antarctica we often talk about how gentoos are doing so well down there versus, say, the chinstrap and adeles, who seem to be, their populations are declining quite rapidly at a lot of the northern parts of the peninsula. Whereas gentoos seem to be increasing and spreading south. So if you talk to some people, they'll often discuss, or they'll put it in terms of who's winning and who's losing against climate change. And part of that, I think, has to do with how gentoos are very plastic animals. You know, they're very flexible compared to adeles and chin straps, and maybe that's what it comes down to in the end, is how flexible these certain species are in Antarctica and elsewhere. So do we have any more questions from the audience? I don't see any hands. Nobody is waving at me. So I think with that, I will close things up and thank the authors for joining us. Thank you, Teresa. Thanks, Alan, for contributing. Thank you, Brendan, as well, for joining, and everyone who came in today. We had about 40 participants in total, which is the most we've had for this time zone so far. So that's fantastic to see some new faces. Divi, do you have any closing comments or anything like that?
No, I'm delighted that we could attract so many people from down under, and that others didn't go back to sleep. Thanks a lot.
Yeah. All right. Thank you very much for everyone. Next week we're back to our European and North American time zone, so we'll be on at 1600 GMT next Wednesday with a couple more papers. And with that, take care, stay inside, wash your hands, stay safe, and we look forward to seeing you again really soon. Bye, everyone.
Bye-bye. Thanks.